All right, let's dive into what's basically a microscopic crime scene. You've got a sample, and it is just teeming with millions of tiny suspects. Our mission? To find the one single culprit responsible for whatever's going on. We're going to uncover the absolutely ingenious techniques that scientists use to isolate, grow, and ultimately identify a single type of bacterium.
So this is the central puzzle, right? A single drop of water, a swab from a patient, it can hold millions of different microbes. How in the world do you pick out just one from that ridiculously crowded party to figure out what it is and what it can do? Well, let's find out. Here's our game plan for today. We'll start with the big challenge. Then we'll get into the classic techniques for separating these little guys. After that, we'll talk about how to feed them, how to use some really smart lab tricks to get them to show their true colors, and finally, how it all comes together to name our culprit.
You know, in the early days, scientists tried a method called serial dilution. It's pretty much what it sounds like, just watering down a sample again and again, hoping that the very last drop would have just one type of microbe. The problem? It was super unreliable. More often than not, that last drop was still a mix. This whole problem really set the stage for a massive breakthrough from the legend himself, Robert Koch.
The whole goal here is to create something called a pure culture. That means you have an entire population of bacteria that all came from one single parent cell. It's like a perfect bacterial family tree, where everyone is an identical twin. And to get there, microbiologists really rely on two main foundational techniques.
The most common method, the one you see everywhere, is the streak plate, and it's just brilliant in its simplicity. You take a sterile little wire loop, dip it into your sample mix, and then you just streak it back and forth across one section of a petri dish that's filled with a nutrient gel called agar. Then, and this is the genius part, you hit the loop with a flame to sterilize it, let it cool for a second, and then you drag it through the very end of your first streak to pick up just a few bacteria, and you start a second streak in a new section. You do this maybe a third or fourth time. With every new streak, you're dropping off fewer and fewer bacteria, until finally you're leaving behind single individual cells all on their own. And boom, this slide just shows you the payoff perfectly. The diagram shows you the pattern, but the photo, that's the real magic. The first streaks are like these crowded bacterial highways, right? But by the time you get to that last section, you see these beautiful, distinct little dots. Each one of those dots is a colony, a visible mound of millions of clones that all started from one single cell that you isolated. You've caught your suspect.
Now, streaking isn't the only game in town. There's another approach called the pour plate method. With this one, you mix a diluted sample directly into warm liquid agar before you pour it into the dish. As the agar cools and solidifies, the bacteria get trapped in place. Then they grow into colonies both on the surface and actually inside the gel itself. This is really handy for certain microbes called microaerophiles, basically critters that prefer lower oxygen environments and actually love growing inside the agar.
Okay, so you've done it. You've isolated a single bacterium. Now what? Well, you got to keep it alive. You have to feed it. And just like people, different bacteria can be pretty picky eaters. This is where culture media come in. We're talking about creating the perfect bacterial buffet. Media pretty much come in two main flavors. On one hand, you have a defined medium. This is like a super precise chemical recipe where you know every single ingredient down to the exact milligram. But for a lot of everyday work, scientists use what's called a complex medium. You can think of this more like a hearty, nutritious stew. It's made from stuff like beef extract or yeast extract, so it's packed with all sorts of good stuff, but you don't know the exact chemical formula.
And what's kind of funny is how simple the recipe for a complex medium can look. I mean, if you look at this list for a basic nutrient broth, you've only got a few ingredients: peptone, which is just a protein source, some beef extract for vitamins, and then salt and water. That simple little soup is nutritious enough to grow a huge range of different bacteria.
But what if the food could do more than just, feed the bacteria? What if we could design media that act as diagnostic tools? These smart media can actually help us select for or even start to identify our specific microbes right there on the plate. There are three main types. First, you've got selective media. These act like a bouncer at a club. They've got ingredients that stop certain microbes from growing while letting your target organism right in. Then there are differential media, which are like little chemical detectives. They have an indicator, usually a dye, that makes different bacteria turn different colors based on their metabolism, basically based on what they're eating. And finally, enrichment media are like a specialized energy drink for a rare microbe you're looking for, helping it grow from a tiny minority into a population you can actually see.
Let's look at a classic example, McConkey agar. This stuff is a real workhorse because it's both selective and differential. First, its bouncer ingredients stop a whole category of bacteria called gram-positives from growing at all. At the same time, its detective ingredients are lactose and a pH indicator. So if a bacterium that's allowed to grow can also ferment that lactose, it produces acid, which trips the indicator and turns its colony a bright, obvious red. If it can't eat the lactose, the colony just stays pale. So with one plate, you've already weeded out a huge group and learned something critical about the ones that are left.
And the technology just gets cooler. Take a look at this Chromagar plate. It's designed to identify different species of the yeast Candida. Based on the unique enzymes that each species produces, they literally grow in different vibrant colors right on the same plate. You get pink, blue, green. It's an incredibly powerful tool for immediate visual identification.
But creating the perfect buffet isn't just about what's on the menu. The air these microbes breathe is just as important. Just like this slide says, getting the right atmospheric conditions is absolutely crucial. While a lot of bacteria are happy with the air we breathe, others are way, way pickier.
And you know, sometimes the solution is elegantly low-tech. For bacteria that need less oxygen and more carbon dioxide, you can use something called a candle jar. It's exactly what it sounds like. You put your culture plates in a big sealed jar with a lit candle. The flame burns up a lot of the oxygen and kicks out CO2 until it just extinguishes itself. And just like that, you've created the perfect atmosphere for those fussy little microbes.
All right, so let's recap. We've isolated our suspect, we've grown it on the perfect menu, and we've even given it the right atmosphere to breathe. Now it's time for the final step, the big reveal, putting a name to our culprit.
For really rapid identification, especially in a clinical setting, labs often use these amazing all-in-one systems like the EnteroTube you see here. It's a single tube that actually contains a whole battery of different tests, each in its own tiny compartment. It's designed to quickly identify bacteria that cause intestinal diseases.
So how does this thing work? It's actually pretty slick. You take a sterile wire and just touch it to one of your pure colonies. Then, you pull that wire straight through the center of the tube, and in one move, you've inoculated all 12 compartments at once. You let it incubate for about 24 hours, and then you just look at the colors. Each color change tells you the result of a specific biochemical test, like, did it ferment glucose? Did it produce a certain enzyme? Then, you group the results and they get converted into a unique code.
So this combination of positive and negative results generates a unique 5-digit number. You just look that number up in a code book, and bam, you have your ID. In this case, the code 34363 tells the microbiologist that their culprit is Klebsiella pneumonia, a pathogen that can cause things like pneumonia. The case is solved.
So from Coach Simple Agar Plate all the way to these rapid multi-test systems, we have developed some incredibly powerful tools to catch and name these microbial culprits. But this leaves us with a really fascinating final question. These techniques are amazing for the microbes we already know how to grow. But scientists estimate that we've only successfully cultured a tiny, tiny fraction of all the bacteria that exist on Earth. The great microbial mystery is far from over.